Okay, so welcome back to返學 DSE, and we are now in biology chapter 26, from basic genetics to molecular genetics. That is the explanation after the di-R, Psi-R, and the di-A, Psi-A. So what is the rationale behind all those symbols? So from DNA to proteins, first of all, we understand the role of DNA first. DNA is the genetic code which determines the amino acids in a polypeptide, which then controls the function of a protein. So how this actually works? So the DNA is read as a 3 consecutive base to form a 1 amino acid. They call the triplet code. For example, ATG on the DNA means that you are going to code for methionine as a amino acid. And there is a table provided in your textbook, which means that all codes except 3 codes can code for amino acids, and each code for 1 amino acid only, while some codes may code for the same amino acid, which means that they are degenerated. So this means they are like 1 or 2 code for the same amino acid, let's say A. And there is only 1 code for the start, that is ATG, and then there are 3 codes for stop. When they are reading the triplet code, there is no gap in between, and we are reading it in a non-overlapping manner. And the code of the same amino acids is universal for all organisms. That's why the term universal is also applied in here. This is a characteristic and a features of the acid. And then from gene to protein, we undergo 2 steps. That is from the transcription to translation. For transcription, it is from the DNA to mRNA, and the mRNA will leave the nucleus, and then the DNA stay inside the nucleus. And then after the translation, the mRNA will then translate it to the polypeptide. So how transcription can work? So using the DNA as a template, there's that we call the template string, and another string is called the coding string. So here this is the template string, and then using the template string, we can synthesize the mRNA. Okay, you may notice that for the mRNA and the template string, the nucleotide, they are complementary. This means that in here is A, while this one is T, and this one is U, this one is A. Because for RNA there is no T, only U. This is the complementary part. And then the mRNA are prepared by the RNA polymerase. Then the 3 ribonucleic acid will attach to the template string as shown there in the previous slide in a complementary manner. It's AU and CG. And then the mRNA is complementary to the template string, while it is similar to the DNA coding string. Remember this term, similar. So what is the difference between mRNA and also DNA coding? Now we have the mRNA. So what happens next is the translation from the mRNA to protein. And then the protein is prepared with the help of rRNAs and also the tRNAs. For mRNA, the long term is actually for the messenger RNA. It is carrying the information from the DNA to the cytoplasm. And then for rRNA, it is the ribosomal RNA. It combines with the proteins to form the ribosome. As for the tRNAs, it carries the amino acids to the ribosome, is the transfer RNA, and the tRNA is complementary to the information on the mRNA. So at this moment, let's have a look comparing the transcription and translation based on their location, mediation, template, and the complementary base pairing, and the input and output. Please complete the table shown in this slide first. Okay, now let's have a look at answers. So for the transcription, it happens in the nucleus, while for translation, it happens in the cytoplasm. For transcription, it's mediated by the RNA polymerase, while for the translation, it's mediated by the ribosome. And the template used for transcription is DNA, while for the translation, which is the later step, is the product of the transcription, that is the mRNA. And then for the complementary base pairing is between the DNA and the 3 ribonucleotides, while for the translation is between the codon and the non-anticodon. And then the input for the first one is the ribonucleic acid, and then the output is the mRNA. And then for the translation, the input is the 3 amino acid, and then the output is the polypeptide. And proteins are made by the ribosome. The ribosome can be found on the rough ER, or they are free floating in the cytoplasm. But either in both cases, they can work perfectly. At this moment, you may wonder what is the difference between ourselves? Because In our normal somatic cells, that is for the cells that commonly found in your body, except your sperm and also the ovum, they all carry the same DNA. However, the gene expression governs their differences. That is, not all the genes are expressed. When different set of genes are expressed, you will have different set of proteins produced, and then it will allow the cell will have different functions. So that's why your cells in the eye is not the same as the cells in your fingers because of gene expression problem. So what actually is a mutation? Mutation is a change of the base sequence of the DNA. So it's introducing a new alleles to a population. So here are the example how gene mutation can actually occur by substitution, inversion, deletion, and insertion. These are the four main reasons of causing a gene mutation. So which of them will have a bigger problem? Okay, let's have a 3-second thought. Okay. In order to know the effect, you have the concept of the reading frame first, because the DNA is actually read as a codon, that is a triplet, and the deletion or insertion will cause frame shift, and then the protein are properly non-functional. Here is an example. ATG is the first reading frame, shown that in red color, okay, non-overlapping manner, okay. Another method to read it is using the green one, and the third method is using the yellow one, while the fourth one is the same as the first one. So we only have 3 methods to read it. So reading frame 1, 2, and 3. And by inserting an extra nucleotide, as shown that in here, from the normal to mutation, using an insertion, and then the whole reading frame is shifted because each time it should read as the red one. And now the red one encodes for the ATG, which is the start of an amino acid. However, in the case down here, there is no more ATG. So there is no proteins produced for read. While the proteins may be produced using this particular reading frame and of this gene. So this gene results in a loss of function. So you may notice that the deletion and insertion will cause more problems than the other one. Other than the gene level mutation, there are also chromosomal mutations. The chromosomal mutation means that there is a structure or number change of the chromosome. So here are some examples: the deletion, duplication, inversion, and also translocation. In particular, we will talk about the Down syndrome, because it's a disorder caused by an extra chromosome 21. So this is a must-taught example in your curriculum. Okay, so how actually mutation, chromosomal mutation may occur? It usually occurs in the homologous pair of chromosome or the sister chromosome fail to separate, and then it results in a non-disjunction. So what is the cause of mutation? Indeed, there are spontaneous mutations, which means it occurs slowly and randomly. However, the rate is usually in a very low rate, so normally it will not affect us. However, as we go older, the mutation accumulate, and that's why for old people, they may suffer from cancer more likely than the young people. And there are also induced mutation. So induced mutation are caused by mutagens. So what are mutagens? So in the case of Spider-Man, the mutagen is spider. However, it's not included in your syllabus, so don't say spider, okay? Even the Spider-Man may scream. So what are the mutagens? So the chemical way is the nitrous oxide, tar, acetal, and the nitric gas. They will change the chemical structure of the DNA and may fundamentally change the protein form. Other than chemicals, radiation will also cause mutation, like the UV light, X-ray, and also gamma ray. Here is an example of how gamma ray may affect a person. True story, right? And also how it can be done, because the ionized water and other molecules will form the free radicals, and then because they are highly reactive, they may damage the DNA molecules. However, in normal case, usually if you suffer from the radiation, you will not become Hulk because you will simply die. Okay, this is not the case in the mouse. Here is the effect of the mutations. Many mutations have no effect on the organism because, like, they occur in the non-coding region. We have the one gene, one protein hypothesis, and then we are studying organisms or their genomes based on their coding regions. That is, they code for a functional protein. And however, in our genome, there are so many regions which is non-coding region, which means it's not code for any functional protein. So if there is a problem over there, so nothing actually happens. And it's also possible that the new code may actually code for the same method because of the degenerative natures of the codon. And also the change in the amino acid may not alter the shape of the protein, because at the end of the day, the shape of the proteins is the one which really matters. And also, it is possible that the effect may be masked by the normal allele, which is a dominant allele. Okay. However, but if something really happens, it is usually bad. Okay. But what is the rationale behind? Let's have a thought about it. And for the mutation and inheritance, so how you may inherit a mutated gene from your father or mother. So for mutations in somatic cells, it is not passed on to your offspring. However, but if the mutation is occurring in the gamete producing cells or even on the gamete itself, it can pass on from generation to generation. Normally, it is a good thing because it will provide a source of variations to the populations. However, because a lot of mutations are not useful, okay, in a human sense, so it may not be that helpful. So if you would like to know more about transcription and translation, please go to YouTube and look for the video from DNA to protein in 3D, and it also has a subtitle in Chinese and English. So here is an extract from the HKEAA about the concept in the molecular biology. So in molecular genetics, you have to understand about chromosome, genes, and also nucleic acid. And the basic two process is transcription and also translation. So after that, you have to know about the Down syndrome and also sickle cell anemia, because sickle cell anemia is an example of the gene mutation, while the Down syndrome is an example for the chromosome mutation. And in the next chapter, we will talk about more about the biotech behind. So this will be the end of chapter 26 intensive videos. I hope to see you guys next time. Bye bye.